everyone, and welcome to another live stream, Histology 101 with Dr. Jamie Chapman. That's me. Today we're going to be continuing our look at the reproductive system. Today we're moving away from the reproductive tract of people assigned female at birth and to the people assigned male at birth. So welcome along. It's great to have you here for those of you who are here. Firstly, let me just also say thank you very much to Dev Eliper, a very interesting name. Welcome along to our followers list, our slide box, as I like to say. When you're a new follower, we say you're a new slide to our slide box, so welcome to our slide box. It's great to have you there. So we're going to start off by discussing spermatogenesis, and then having a look at the histology of the testis, and then the various other ducts, so the intra and extra-testicular ducts, the ductus deferens, the seminal vesicles, accessory sex glands, so the seminal vesicles, the prostate gland, and then looking at the structure of the penis and so on. So we'll get started, I think, by having just a very quick discussion about spermatogenesis, if that's okay, unless anyone has some specific questions. And then we'll move on to looking at the finer histology of the testis and moving forward. All right, so here we are. I'm just going to grab my drawing tablet. Okay, so we're going to talk about spermatogenesis. Of course, that's the production of sperm, and of course this takes place within the seminiferous tubules of the testis after puberty. So before puberty, the only things which sort of reside within the testis are stem cells, which are Spermatogonia. So a single is a spermatogonium and multiple are spermatogonia. And we also have, housed within there, we have our nurse cells, which are also known as Sertoli cells. So from our initial spermatogonium, this is our stem cell. So it's a diploid cell. These undergo a number of mitotic divisions and produce lots of different spermatogonia. There's various subdivisions. Now, I don't talk about the various subdivisions. We often describe things like pale A spermatogonia and type B spermatogonia, and type B eventually become the next phase of the meiotic division, or the first cell to undergo meiosis, and that's the primary spermatocyte. Now, I'm not going to go into that level of detail. That's not really what this is all about. Connie Woodward, thank you very much for the follow. Welcome to the slide box. It's great to have you along. So our stem cells are our spermatogonia. Eventually, one of these will differentiate. We get sort of mitosis. One of these will stay behind as our stem cell, and one of these eventually will move on to eventually differentiate and eventually become our primary spermatocyte. So this will still remain behind as a spermatogonium. Now, the primary spermatocyte is also a 2N or a diploid cell, 46 paired chromosomes, and 23 pairs of chromosomes, I should say, for 46 in total. And now the primary spermatocyte, because it's the cell which first enters into meiosis, enters into sort of duplicating its chromosomes, so that we know that a normal sort of chromosome has a single sort of chromatid-like structure, but then during the S phase of the cell cycle, it duplicates that chromosome, so it forms this duplex chromatid type of chromosome. So this is a chromatid. So that's a chromatid there, and so this structure is known as a double chromatid chromosome. But in fact, it's really two chromosomes stuck together by this structure called the centromere. Will we need to go into that much detail for our upcoming exam, Tarini asks. I'm not sure about what level of detail you mean about the spermatogonium, you know, the differentiation and so on. You won't need to go into any detail about that. How exactly do nurse cells aid spermatogenesis, Histolover asked. So we'll get there, we'll talk about that, but the nurse cells regulate spermatogenesis through the stimulation of follicle stimulating hormones. So they actually have receptors for FSH, and then that actually helps to drive spermatogenesis. So they actually physically push the sperm through the various compartments and regulate the release of the sperm at spermiation when they're released into the seminiferous tubules and so on. So we'll hopefully talk about that. I do want to draw out a diagram of the seminiferous tubules, talk about the blood-testis barrier and so on. So as I was saying, the primary spermatocyte enters into the S phase of the cell cycle, so it duplicates its chromosomes. We still say it's 2N, but those chromosomes that it has are these duplex or double chromatid type of chromosomes. So the primary spermatocyte undergoes meiosis 1. So meiosis 1, of course, is where we get things like the crossing over in prophase 1. So the cells that are actually made during this process are actually genetically different from the person within which they're made, because parts of the homologous pairs of chromosomes, so remember we've got 23 pairs for a total of 46 chromosomes. Now 23 of those chromosomes are chromosomes that you got from your mother's oocyte, and 23 that you got from your father's sperm. So we've got maternal and paternal chromosomes. And they're referred to as homologous chromosomes. Don't know if that hasn't all fit on the screen. Let me just... So we actually number them 1 to 23, and so they eventually line up during meiosis 1. They pair up. So chromosome 1 meets chromosome 1, and chromosome 12 meets chromosome 12 for the full 23 pairs. And this all happens during prophase of meiosis 1. So they all align along the metaphase plate, and eventually those pairs of chromosomes separate. Now that arrangement of those chromosomes, maternal, paternal chromosomes side by side, is a random assortment. So we have 2 to the power of 23 different variations of cells that can be produced through meiosis 1, which is about 6, I think it works out to about 6 billion, something like that, or 6 million, sorry, 6 million different variations of cells that can be produced during meiosis 1. You're welcome, Histology. So during meiosis 1, primary spermatocytes undergo cell development, and they produce 2 cells which are secondary spermatocytes. Now these are haploid because they only contain 23 chromosomes, but again they're still of this duplex chromatid type of chromosome. Okay, so we still actually got 2 chromosomes stuck together, but there's only 23. So whereas up here we had 46 of these paired or double chromatid types of chromosomes, the result of meiosis 1 results in 2 secondary spermatocytes, each with 23 chromosomes, so they're haploid. This is diploid, or 2N, haploid or N, but they're of this duplex chromatid type of chromosomes. So these still need to undergo meiosis 2, a second meiotic division. So these secondary spermatocytes immediately enter into meiosis 2, and each of these 2 cells divides and produces 4 cells, which are known as spermatids. Now these are N, and these are now the single chromatid type of chromosomes. So these are N, these are haploid. That's really bad writing, I apologize. So that's pretty much it for meiosis. These spermatids, of course, when they actually divide, they're actually just little round cells. But so they don't actually look anything like the normal structure of the sperm that we sort of come to know, which is this structure here. This is the spermatozoon. Okay, so this cell, these round spermatids, then need to actually mature, undergo this morphological transformation from this round spermatid cell to this mature spermatozoon that's eventually released into the lumen of the seminiferous tubules. Now, this process is a process called spermiogenesis. So spermiogenesis is this morphological transformation of these round spermatids into the mature spermatozoon. So it's got its head, it's got its acrosome, it's got its midpiece with all of its mitochondria housed within here, which is powering the flagellum, enabling that sperm to swim those long transits through the reproductive tract of the person assigned female at birth. And then it's got its flagellum, which of course is a sort of a cilia-like structure containing 9 pairs of peripheral tubules, 2 central pairs, and they're powered by the motor protein called dynein. And so they need to get the energy somehow, and we'll talk about the contribution of the seminal vesicles and the prostate gland in providing energy for our mitochondria within our sperm for that long journey. So spermiogenesis sounds like, yeah, good watermelon, yes. I did make a joke in my lecture that I had a GIF that I created of the transformation of a round spermatid to a spermatozoon, and I made the joke that it sounds like a transformer when it transforms and then becomes a spermatozoon.
This is not my final form.
All right, this is its final form. This is the spermatozoon. Now, when the sperm are actually released, that's a process driven by our nerve cells, and that release of the sperm into the lumen is known as spermiation. So spermiation is the release, sort of the breaking of the cellular junctions of these spermatozoa into the lumen of the seminiferous tubules, and that's the process of spermiation. So this whole process is, of course, spermatogenesis. So this is what we've been talking about, spermatogenesis. We've got mitosis of the Spermatogonia. Annoyed Patty, hello from Uruguay. Thanks very much for coming along. Everyone, Annoyed Patty I met on Twitter a number of years ago, and she's a med student from Uruguay, and she was having some issues with histology, wasn't having a lot of fun, so I sent her a link to your CAM 101 histology iBook, and hopefully that helps her out. I know not a lot of people find histology very useful, and so we sort of become Twitter friends because of that, and I've just been very inspired by her journey as a medical student in Uruguay. So yeah, welcome along. I'm really glad you could come along. She long past histology, thank goodness. She doesn't have to deal with it anymore. Yaminki, marijuana is legal in Uruguay. Well, that's good for some. Apparently it's going to be legal in Canberra too, which is the capital of Australia. For those of you from overseas, that'll be the first, well, it's not a state, it's actually a capital territory that's actually going to legalise marijuana for personal use, as far as I understand. Anyway, that's a little bit off topic. We can talk about how smoking damages cilia and therefore may affect the reproductive system, and in fact sperm. Sperm, just like cilia, can be damaged from smoking, so it can affect fertility. So anyway, that's a little bit of a side issue there. So this is spermatogenesis. We're talking about the mitosis of the Spermatogonia, the differentiation of one of those spermatogonium to eventually become a primary spermatocyte, which is diploid. It's that cell which undergoes meiosis 1 to produce 2 cells, 2 secondary spermatocytes. They're haploid, but of these duplex chromatid type of chromosomes. They immediately enter into meiosis 2, producing 4 spermatids. Whoops. I'm being quite careful with my pen here. I'm trying to be anyway, because yesterday I deleted a drawing that I did for those of you who weren't here, which was a little bit heartbreaking. So we produce 4 spermatids, and those are all haploid cells, 23 chromosomes, single chromatid type of chromosomes, numbered 1 to 23 of those chromosomes, and then it undergoes the morphological transformation through a process called spermatogenesis to produce the spermatozoon, which are then released into the lumen of the seminiferous tubules at a process called spermiation. So basically that's an overview. Now, one thing I haven't really sort of talked about, when we actually get division of these cells, something relatively unique happens in the male, is that when we actually get this meiotic division, we actually get what's called incomplete cytokinesis. So normally in mitosis and meiosis, we actually get complete separation of the cells. You know, we get that little cleavage furrow, we get the microfilaments forming, and then eventually the two cells separate completely. That's known as cytokinesis. Well, in meiosis, in males, in people who have testes, they actually don't actually completely separate. They're actually connected by these little stalks. So we have our cells here, and they're connected by this little cytoplasmic bridge. And so when we actually produce our 4 cells, they're actually all sort of held together by these little cytoplasmic bridges. And so that's unique to the male compared to what we looked at in the female. So let me just allow these. I'm sorry. Twitch really doesn't like reproduction, doesn't like anything to do with sex. It took me a long time to actually come up with the title for this, because I can't use the term testis, I can't use the term sex, I can't use the term gonad apparently. So any comments which are coming in the chat may be modified by Twitch, which I then have to release. So Ruby's asked, spermiation is the release of spermatozoa into the lumen of the seminiferous tubule, driven by nurse cells. That's correct. No worries, it came up twice, probably because you didn't realize that it went through the first time, and it holds it for moderation, and I actually have to click to release that comment. But yeah, that's exactly right. So spermatozoa spermiation releases sperm into the lumen of seminiferous tubules driven by the nurse cells. Yeah, that's exactly right. So we were talking about this cytoplasmic bridge. It's unique to male meiosis. We don't see it in female meiosis. We have that weird sort of different things that happen in female meiosis that we talked about, you know, the first polar bodies and only one viable cell. So that's one of the unique events in the male, is this cytoplasmic bridge. Now, spermiation actually involves snapping these bridges during this maturation, part of this spermatogenesis process, as well as snapping these bridges and then having these single cells. Now, it's suggested that the reason that we have these cytoplasmic bridges is it's to regulate the release, coordinate the release of X and Y bearing sperm, so that they're not released in different waves. I don't know how true that is, but certainly that's one of the suggestions that has come up. All right, is there anything else I want to talk about this? And we can begin to have a look at the structure of the seminiferous tubules. Does anyone have any questions about this before we move on? Okay, so I might just save this. I don't know if we'll ever need to come back to it or not, but we can come back to it if we need to. So I will clear this now. All right, so the seminiferous tubules, of course, is the location where spermatogenesis takes place. Now, it sits on a basement membrane there, and then on the outside, let's do this color. I'll just increase the size here. And on the outside of the seminiferous tubules, we have several layers of cells called myoid cells. And they're cells which undergo contraction to help move the sperm through the seminiferous tubules. So we've got these myoid cells on the outside here. These are sort of like contractile-like, similar to smooth muscle cells, but they're not actual smooth muscle cells. So you just refer to them as myoid cells. Okay, now, so sitting on the basement membrane is our nurse cell, and the nurse cell extends all the way from the base, sitting on the basement membrane, all the way to the lumen. So what I'm going to do is draw a couple of these here, like so. We'll put a cell or two in here. These aren't eyes. It looks like someone's peeking out, hiding underneath the Sertoli cells. So we have these cells here. Just bear with me. This is going to take just a little while. Okay, so we've got another cell here. So we're just going to fill in, imagine that one there. Okay, and the nucleus of the Sertoli cell typically sort of very sort of pyramid shaped, sort of triangular shaped, with the apex of the triangle always pointing towards the lumen. So this is the lumen of the seminiferous tubule. This is the outside. So again, we're looking at the seminiferous tubule here. Okay. Now in this region here, I might just add a different color here. We actually have lots and lots of tight junctions, and this part here, this is actually the blood-testis barrier. So these are tight junctions, or zonular occludens, held together between adjacent nurse cells. So So these are nurse or Sertoli cells. I've used both terms here. Sertoli is what we call the eponymous term. Nurse cell is the non-eponymous term that we give to it. So housed within here, we actually have our primary spermatocytes, and then over here we'd have our secondary spermatocytes. Over here we'd have our round spermatids. And then over here we've got our sperm with their tails sort of poking out into the lumen. I'm just going to draw these quickly because I think you can get the idea. Okay, so this blood-testis barrier serves a couple of purposes. One, remembering, so when we talked about meiosis 1, what happens is we get those crossing over event that takes place during prophase 1, where parts of the maternal and paternal chromosomes mix, meaning that the cells that are produced after meiosis 1 are genetically unique from the person within which they're being produced. What that means is that they're genetically foreign, so that could be potentially identified as foreign by the person's immune system. And so to protect these cells, particularly the secondary spermatocytes onwards, we actually have this blood-testis barrier which sort of prevents the immune system from being able to identify and reach these cells, these new genetically different cells. The other thing that it does, it actually creates two compartments within the seminiferous tubules. So this compartment here is known as the basal compartment. Just checking that my head wasn't in the way. The basal compartment, and housed within the basal compartment is where we find our Spermatogonia. So the Spermatogonia are always sort of sitting on the basement membrane here in the basal compartment. All of these cells pass the blood-testis barrier. This is a compartment known as the adluminal compartment. So here we have our primary spermatocytes, we have our secondary spermatocytes, we have our spermatids. And then here's our spermatozoa, sort of sitting close to the lumen here. So housed within the abluminal compartment, we have our primary spermatocytes, we have our secondary spermatocytes, and we have our spermatids. So when these Spermatogonia undergo mitosis and one of those differentiates and becomes a primary spermatocyte, part of that maturational process involves pushing past this blood-testis barrier and to move from the basal compartment into the adluminal compartment. And then that primary spermatocyte enters into meiosis 1, produces 2 secondary spermatocytes. They both enter into meiosis 2, producing 4 spermatids, and then spermatogenesis results in these 4 sperm on the surface there. So that's pretty much the structure of the seminiferous tubule. So we can actually begin to have a look at the structure of the testis in a little bit more detail now, unless anyone has any specific questions about anything I've drawn there. Does that mean during spermiation the cells have to push past the tight junctions one last time? No, spermiation actually involves the release into the lumen out here. So once the only cell that needs to push past is that cell that becomes the primary spermatocyte. So these are just released into the lumen. They're generally separated from the immune system there because of the tight junctions. And then during the rest of the tract, they may actually interact with the immune system and so on, but it's not a very common thing for them to take place. Is the blood-testis barrier visible on light microscopy? No. So because it's tight junctions, those are zonular occludins. Those are cell membrane, transmembrane proteins holding the two membranes together, like a little stitching that happens between the two cell membranes. So it's an electron microscopic, only something which you can see with the electron microscope. You won't see them with the light microscope, because cell membranes are too small to actually resolve with a light microscope normally. So the tight junctions make the blood-testis barrier just at that bottom part. That's certainly my understanding, is that the blood-testis barrier is about separating these self cells, because these are still genetically identical to the person within which they're being made. It's only after prophase 1 that the cells potentially become genetically different. So the blood-testis barrier only separates those cells in the basal compartment. They're fine. It's only those in the adluminal compartment which need to be separated from the immune system. So histo 1, 2, 3, 4, 5, 6, 7, 8, 9. Hopefully that answers your question there. All right, those are really good questions. Thanks very much for posting them. It's really great to have you along and interacting and getting some of the answers that you have questions about. All right, so you're welcome. We might now move to the histology of the testis and beyond. So I'm going to switch over to the digital slide box. So I'm going to start off with this Trichrome stain section of the testis, and this is actually, it's one of my favorite slides. I think it's really beautiful because it highlights the different components of the testis. Blue is collagen, of course, and one of the things about the testis is it's surrounded by this very dense connective tissue coat called the tunica albuginea. And so that's on the outside here. And then the tunica albuginea throws in these connective tissue septa to divide the testis into lobules. And within each lobule we have 2 to 4 seminiferous tubules, which are sort of blind ending tubules within the testis. After you drew it out, I found it really useful having a schematic diagram like this before going straight into the actual histodiagrams in the lectures. At least I know what I'm looking at. I'm glad that helps, because it can be a little bit complicated to work out where everything is taking place there. Great. All right. So we've got these nice connective tissue septa. We can see how thick this tunica albuginea is. It's dividing the testis into the lobules, and then these little holes here, these are actually the seminiferous tubules. And so these are blind ending tubules, and you can see they're highly convoluted, so they fill up a lot of that space there. So I'm just going to keep zooming out for a second while we continue our description, low magnified description of the testis. We see this thickening of the connective tissue in this region here. This is known as the mediastinum of the testis. And housed within it are these little tears, and these actually look like little tears in the connective tissue. But in fact, when we zoom in, we can actually see that this red here, this is actually an epithelium. It's lined by an epithelium. So this is part of the intratesticular ducts known as the Rete testis, R-E-T-E testis. And it's part of the intratesticular duct system. And we'll talk about what's going on here in a little while. All right, so this is the mediastinum of the testis. This is the tunica albuginea throwing up the septa. This is the spermatic cord. And here, just on the posterior surface, we've got a little bit of epididymis, which we'll talk about at a later stage. Now, if we zoom in, we can actually, once we wait for the virtual microscopy to catch up, there we go. See thick, dense connective tissue collagen here, and then you can see there's some blood vessels on the side closest to the seminiferous tubules. Now, we can sometimes subdivide this layer here into what's called the tunica vasculosa. So this inner part where the blood vessels are is sometimes referred to as the tunica vasculosa. So here we have the tunica albuginea, then the tunica vasculosa. And then we've got our seminiferous tubules here. You can see the collagen that's forming part of the wall of the seminiferous tubules. There would be myoid cells here as well. So the testis then, which of course is the gonad within people identified as male at birth, has two major functions. One is the production of the gametes in the form of the spermatozoa, and the other of course is the production of the sex hormones, largely androgens, things like testosterone and dihydrotestosterone and so on. Now, the sperm, of course, are formed within the seminiferous tubules, whereas the sex hormones, the androgens, are actually formed by cells out in the interstitium. So this space in between the seminiferous tubules is known as the interstitium of the testis, and housed within there we have interstitial endocrine cells, or cells known as Leydig cells, or interstitial endocrinocytes. And so they're always associated with lots of blood vessels. So the seminiferous tubules are producing the sperm, the interstitial tissue, the interstitial endocrine cells are producing the androgens, and so that's how together they function as the gonad of the male. Now, this trichrome stain section is really good for staining the collagen and differentiating between the different components of the testis. However, it's not very good at morphology of spermatogenesis and the structure of the seminiferous tubule. So it's a nice sort of overview. You can see the connective tissue septa here. Here's the mediastinum. We can see the blood vessels here. This is part of the pampiniform plexus of veins. This is one of those components that helps to regulate temperature of the testis. So we've got a number of different mechanisms which help to regulate temperature in the testis, because spermatogenesis requires about 2 to 3 degrees lower than normal body temperature for normal spermatogenesis. And there's a number of ways it can help to control that. Leydig cells produce testosterone equals ladies like testosterone. That's how you remembered it. Well, whatever helps you remember. There's always little things like that that helps me remember things as well. So the mechanisms for maintaining temperature, we have the smooth muscle, or we have the muscle within the skin of the scrotum, which is known as the dartos muscle. That can contract when cold and relax when warm, so that we can increase the surface area of the skin for evaporation. We have the cremaster muscle, which is a muscle which exists within the spermatic cord, and that can raise and lower the testis as well, according to temperature. And then we have the countercurrent heat exchange that occurs within the spermatic cord here. So the testicular artery, you can see a nice artery here, or the spermatic artery. So you can see lots of smooth muscle here. You can see that little wrinkly internal elastic lamina. So this is the spermatic artery, and then we've got the veins out here. We have this sort of countercurrent heat exchange, so where the scrotum has been cooling the blood on the surface through evaporation, so that venous blood is cool. The warm arterial blood that's coming in bypasses the cool venous blood, and they get this exchange happening whereby the incoming arterial blood is cooled and the exiting venous return is actually warm, so we get this countercurrent heat exchange. And so these three mechanisms help to lower the temperature of the blood within the testis, or the temperature within the testis for normal spermatogenesis. You can see lots of adipose tissue out here, but you can see how many and how complex these blood vessels are in this spermatic cord. And then here we've got the epididymis, which is just gorgeous. Histologically, it's the most pretty epithelium in the human body as far as I'm concerned. Very regular epithelium, these long stereocilia on the surface. Look at these. Anyway, we can talk about the histology of that another time. We'll get to that. All right, so now we're going to have a look at the structure of the seminiferous tubules in a little bit more detail. So I have this really nice resin section, and whenever we have sections of resin, they're only about 1.5 microns thick, whereas the normal paraffin section is about 5 to 7 microns thick. So there's less interference, and therefore the resolution is much greater in a resin section. Is there a reason why, of all types of cell division, spermatogenesis is temperature sensitive? I'm not exactly sure. I certainly know that it's the primary spermatocytes which are the most liable to heat damage, which can then affect the later stages of spermatogenesis. I'm not sure why, in particular, this type of cell division is under so much temperature regulation. I'm not really sure. Mediastinum is thickening of tunica albuginea. It's a thickening of the connective tissue. I don't know if we consider it a thickening of the tunica. Well, it certainly looks like a thickening of the tunica albuginea. We sort of got a meet up here from the outside to the inside of these stroma, or septa, I should say. But it's certainly an area of thickening of connective tissue. So, I think that's probably a fair thing to say, Ruby, that it is a thickening of the tunica albuginea. So as I was saying, this is the resin section, so we can actually identify the cells within the seminiferous tubules much more clearly. You can see this thick connective tissue of the tunica albuginea. You can see the blood vessels of the vasculosa layer, and then if we begin to zoom in here, we can actually see a lot more of the fine cellular detail. Now, this is a section of the human testis. So we can actually look at the different cell types that we find within here. All right, so we can see the thick wall of the seminiferous tubule. We can see the myoid cells. These are the nuclei of the myoid cells, so these are the contractile cells. When the sperm are first released into the lumen, they're unable to swim on their own, so there's a fluid which is secreted by the nurse cells, and they actually called testicular fluid, and so the immotile sperm are in the testicular fluid, and it's the contraction of the myoid cells that helps to move them through those duct systems. So here we begin to try to identify the different cells in this section. So one thing to look out for when trying to identify nurse cells or Sertoli cells is to look for a really prominent nucleolus in a relatively large nucleus, often quite basally located, but again, looking at the shape, you can notice how this one here is quite pointy. A lot of the time, the nurse cell nuclei are quite pointy, and they have a prominent nucleolus. This one, obviously, we're not through the section of the nucleolus, and this one we are. So if you see something like this, these are the nurse cells within the testis, within the seminiferous tubules. So anything with this nice big nucleus, prominent nucleolus, is going to be a nurse cell. Okay, so remembering they extend all the way from the basement membrane all the way to the lumen there. Now, any of these round cells which are sitting on the basement membrane, these are going to be our Spermatogonia. Okay, so these are our stem cells. They're sitting in the basal compartment. And then these cells here with the really large nucleus, very irregular in appearance chromatin within the nucleus, these are our primary spermatocytes. So they really look like very large cells, very large nuclei, very irregular appearance to their chromatin. So they often have this sort of, I guess, leopard skin type of pattern to the chromatin here. So these are our primary spermatocytes. You can see lots of them over here, for example. So lots of primary spermatocytes. Our Spermatogonia are sitting on the basement membrane. Here's our primary spermatocytes. Now, we generally don't identify secondary spermatocytes. They're only in the testis for a few hours. So anything beyond these small round cells, these we would probably identify as spermatids. Okay, so these are haploid cells. These are the ones that are going to undergo the division and eventually become the sperm. Now you can see how dense the nuclei are of our developing spermatozoa, and their tails are sort of leaping out into the lumen of the seminiferous tubules here. So what happens is, as part of this spermatogenesis process, the DNA changes. The proteins within the DNA change in structure. So normally it's histones which hold the chromatin together in a nucleus, but in sperm, the nucleus needs to be compacted down into a very, very small space. And so they use a special type of protein called histones. And histones have an increased ability to have disulfide bonding, which really tightly compacts down the DNA. So that you have this very dense nuclear condensation, so you have this very darkly stained nucleus. So out here we've got the interstitium. We can see the blood vessels within the interstitium, and then these very big pink puffy cells, these very acidophilic cells, you might be able to see some of the holes within the cytoplasm. These are our interstitial endocrine or our Leydig cells. So they actually have lots of lipids because these are steroid producing cells. Testosterone takes its origin from cholesterol, and therefore it's steroid producing. And so the typical features of a steroid-producing cell, they have lipid droplets, they have lots of smooth endoplasmic reticulum, and they have mitochondria with tubular cristae. So the cristae is a little bit different in structure to protein-producing, mitochondria in protein-producing cells, because part of the lipid production process involves the mitochondria. What are the white patches between the cells in the seminiferous tubules? You mean here? I'm sure there's some shrinkage artifact that's taking place in this region, so the cells are actually separated, so it's just a little bit of artifact, good watermelon. So again, it's one of those things that we tend to just sort of ignore in this area. So we mainly look at just the nuclear shape, the nuclear staining pattern, and their location. Ignore these spaces here. Well, there's some big Leydig cells or interstitial endocrine cells there. So an interesting thing is that in the human testis, different parts of the seminiferous tubules actually undergo meiosis at different stages. So you can see here we've actually got some spermatozoa. You can see the dense nuclei here, whereas on this side there's very few spermatozoa. So this part of the seminiferous tubule is undergoing a different stage of meiosis to this part is undergoing a different stage to this part, and so on. So we call this actually the cycle of the seminiferous tubule epithelium, and basically what it means is that throughout all of the tubules we have this continual turnover of sperm development. Interstitium is between seminiferous tubules containing endocrine cells and blood vessels. That's exactly correct, Ruby. So you can see these big pink puffy cells. These are our interstitial endocrine cells. Look at all those holes where the lipid droplets would be. These are very large cells, big round nucleus. And then we've got our capillaries in this region as well. So the hormones are being secreted directly into the surrounding bloodstream. Okay, look at the size of these cells. They're big, large, acidophilic cells, lots of smooth endoplasmic reticulum. The holes are where the lipid droplets have been washed away through normal processing for microscopy. They're gorgeous in this section, aren't they? Yeah, so really, those are the major features. Oh, look, we've got a love heart. Anyway, that was just a little bit of distraction there. All right. Does anyone have any questions? Oh, look at those interstitial endocrine cells. They look amazing between those seminiferous tubules there. All right. So that's pretty much it. That's how you sort of identify those different components. Let's find a different one. And let's see if we can identify the different parts again. Just sort of a little bit of revision. All right, so this is a little bit trickier. I would identify this nucleus as belonging to a nerve cell because of the prominent nucleolus, its location just above the spermatogonium here. This one too, this one. Okay, so once you sort of begin to identify the nerve cells, they do tend to stick out a little bit more. So again, the ones sitting on the basement membrane, these are our Spermatogonia. These ones with the large nucleus with a very irregular chromatin pattern, these are our primary spermatocytes. The small round ones would be the Spermatids, and then here we've got our Spermatozoa. So the initial cells, often we still refer to as Spermatids until they reach sort of the lumen, and then they're known as the Spermatozoa. And so the spermatozoa are eventually released into the lumen at spermiation. All right. So I think it's a lovely section. It's a nice one to sort of discuss about the different stages of spermatogenesis there. This is a similar section. This is from the testis of a monkey. Again, it's been stained with resin. I don't think it really shows much different than what we were already looking at. Got some really nice sperm here with the tails leaping out into the lumen here. Again, we've got our myoid cells forming the wall, not as thick as in humans, obviously. We've got our Spermatogonia sitting on the basement membrane here. Here's the nucleus of one of our nerve cells. Here's another one here. And so then we've got our primary spermatocytes with the irregular chromatin. We've got our spermatids, and then we've got our spermatozoa. I'm not sure about the interstitial tissue. We might sort of zoom out a little bit and see if we can... This is quite a well-preserved section, so... So this would be one of the interstitial endocrine cells. We've got some blood vessels over here. It certainly looks a lot more compacted than what we were looking at in that previous section. So again, we've got our interstitial endocrine cells here and our blood vessels. You can see the little red blood cells here. All right, Annoyed Patty, thank you very much for the follow. Great to have you along. I'm glad you could come along to the stream and have a look. So this is a paraffin section. So anything that's not a resin section tends to be a little bit more difficult to identify histologically. So we can still see the thick tunica albuginea on the outside here, and we've got a bit of the vasculosa here, and then you can see the preservation here is really quite awful. So we've got lots of spaces. The cells don't look particularly healthy within the seminiferous epithelium. Certainly it would be very, very difficult and very mean of someone to ask you to identify anything here. The preservation is so poor. So I think we might just skip this slide. I don't think there's anything else of particular interest. I think over here we've got a little bit of the Rete testis. So some of this will be part of the mediastinum of the testis over here. And so if we sort of zoom in, we can actually have a look at the epithelium here. Just see it's lined by an epithelium. It's not just simply tears in this section here. So this is part of the Rete testis, which is sort of the junction between the seminiferous tubules and the efferent ductules. Some blood vessels, again more seminiferous tubules over here. So this will be part of the spermatic cord, I think, with the pampiniform plexus of veins here, and we've got our spermatic artery and so on. So you can see how closely related they are there. All right, so I think again this is another section of the testis. I think this one's from a rodent, a mouse. Now the reason we can tell that is because look at all these lined up spermatogonia sitting on the basement membrane here. That's very typical of a mouse. Now we know we talked about the the cycle of the seminiferous epithelium in humans being in different stages, whereas in mice, they actually have the same stage of meiosis in the entire seminiferous tubule. It's just the various length that may change rather than the same section of the tubule. I don't think we're really going to get much more information out of this than what we were looking at before. So this would have been a nurse cell. So certainly for my students in an exam, I would certainly use a micrograph with labels of the resin section, asking students to identify the different stages there rather than one of these slides. Got some nice interstitial endocrine cells out here, Leydig cells out in the interstitium. Various cytophilic cells here. You can see them here and here. All right. Again, we've got human testis, I think, here. We'll just quickly zoom in. This is a bit better preserved. So lots of profiles of the epithelium here, seminiferous epithelium. Again, the preservation is not that fabulous, so we can't. It does become a bit problematic to identify the different cell types here. But what we might find that this might be useful for is we've got some epididymis on the back there, and we've got some of the spermatic cord, but we're not going to be talking about that right at this point in time. Okay, so this is the epididymis. Where is testosterone made in females? Well, the tunica interna actually produces an androgen, but most of that's actually converted into an estrogen by the granulosa cells in the presence of FSH. So a lot of the testosterone or the androgens that a woman has, which brings about the development of pubic hair and some of those characteristics, comes from the adrenal cortex, the zona reticularis, which is the third of the 3 layers of the adrenal cortex, produces androgens, and so women get a lot of their androgens from the adrenal cortex under the control of adrenocorticotropic hormone from the anterior pituitary. And sometimes women can actually experience a pathology related to that, and there can be an overproduction of androgens by the adrenal cortex, and that's known as androgenital syndrome. It's basically as if the person was actually taking anabolic steroids. So when a woman takes anabolic steroids, it brings about a masculinization of the body. So we get increased muscle mass, we get deepening of the voice, and all the sort of typical secondary sexual characteristics of a male, for example. Often what happens is the clitoris enlarges, becomes almost penile-like, and so on. So that's a pathology that can sometimes happen as a result of a syndrome within the adrenal cortex. All right, so if we think about the pathway of sperm, we've now just sort of talked about the production of sperm within the testis here. It might be worth just sort of talking about the pathway of sperm through the rest of the duct system here. I might just quickly grab my drawing tablet again. So of course, we start with the seminiferous tubules. Then the seminiferous tubules sort of finish as this small region known as the straight tubules. That then becomes the Rete testis, which then flows into the efferent ductules. The numerous efferent ductules eventually coalesce into one Ductus epididymis. The Ductus epididymis becomes continuous with the Ductus deferens. That then joins on with the duct from the seminal vesicles. So we've got our seminal vesicle. Whoops. Might help if I could spell. So they join together to form the ejaculatory duct. Which empties into the prostatic urethra, goes through the membranous urethra, and then via the penile, penile or the spongy urethra. So this is the pathway of sperm. They're made in the seminiferous tubules. They then go via the straight tubules to the Rete testis, the efferent ductules, the Ductus epididymis, the Ductus deferens, joins with the duct from the seminal vesicles to form the ejaculatory duct. Now this is within, so this is within the prostate gland. It then passes through the urogenital diaphragm, through the membranous urethra, and then within the corpus spongiosum. In the penile spongy urethra. So of these ducts, these are the intratesticular ducts. And then these are all considered the extra-testicular ducts, outside of the testis. So these would also be considered part of the extra-testicular ducts. So the important processes that sort of take place, these are sort of course, where spermatogenesis takes place. There's not a lot that happens in these regions. The efferent ductules are mainly responsible for resorption of testicular fluid. So most of the testicular fluid is resorbed by the efferent ductules. The ductus epididymis is really important for post-testicular maturation. So it's actually where within the ductus epididymis that sperm gain their ability to swim. So they actually gain their forward motility within the ductus epididymis. They're stored within the ductus deferens. And then during ejaculation, we get sympathetic contraction of the muscle wall of the ductus deferens, even part of the tail of the ductus epididymis. During the emission phase of ejaculation, it moves the sperm into the ejaculatory duct. We get contraction of the smooth muscle in the seminal vesicles, again by the sympathetic nervous system during the emission phase of ejaculation into the ejaculatory duct. We get contributions from the prostatic glands, through contraction of the sympathetic nervous system, the smooth muscle there, and then we get semen formation, and then we get ejection through the opening in the external urethral orifice. So that was messy, but hopefully made a little bit of sense about the pathway of the sperm through the male reproductive tract, or the reproductive tract of people identified as male. Yeah, so we're going to actually have a look, we'll quickly have a look at some of these ducts now. I don't have sections of all of them. I've got a picture of the efferent ductules, but I don't actually have a virtual microscopy slide of it, I'm sorry. And then we can have a look at the epididymis, the ductus deferens. We can have a look at the seminal vesicles and the prostate gland and the urethra there, and then finish off having a look at the penis. All right, any questions about the pathway of sperm through the reproductive tract of people identified as male? All right, if there are no questions, then I'm just going to put this away and we can have a look at some of these sections. All right, so I might just save that. And close it. All right. So we talked about the pathway from the seminiferous tubules to the straight tubules to the Rete-testis, and we can actually see some of that here. It's actually quite nice. So here we can actually see the seminiferous tubule epithelium. Rubias, ductus deferens store sperm and contract with SNS stimulation. That's exactly right. So humans primarily store the sperm within the ductus deferens, and then during ejaculation, ejaculation is divided into two phases. It's an emission phase and an ejection phase. The emission phase is responsible for moving all of the sperm and the seminal fluid into the urethra in preparation for the ejection phase. And that emission phase is brought about by the sympathetic nervous system. So the parasympathetic nervous system is responsible for erection of the penis. Sympathetic nervous system is responsible for the emission phase of ejaculation. And then there's a lumbar reflex that happens for the ejection phase that causes contraction of the bulbospongiosus and the corpospongiosus muscles. Ischiocavernosus muscles, and then that forcefully ejects the sperm out during the ejection phase of the ejaculation. Good Watermelon asks, does the blood-testis barrier extend all the way from the seminiferous tubules to the end of the urethra? No, I don't think it does. Certainly we only describe the blood-testis barrier in the testis, in the seminiferous tubules. There may be things that happen along the ducts that may separate the sperm from the immune system, because we do know that producing antibodies against your own sperm can affect fertility, and certainly that's one of the reasons why a person who has a vasectomy may actually have difficulty after reversal, because often what happens is during the vasectomy, you cut the ductus deferens, you tie off either ends, sperm are still produced and they still reach the proximal end of that cut ductus deferens. It may increase hydrostatic pressure and rupture. That gives the immune system access to the sperm, and the male begins to produce antibodies against his own sperm. So that if that is then reversed, oftentimes the sperm may actually have antibodies stuck on their surface, which interferes with the sperm's own molecules, which are important for them to actually bind to the oocyte egg coats and eventually cause fertilization. But I don't know what sort of modifications there are to the rest of the duct tract that may separate it from the immune system. It's a really good question, and probably something I'll have to look into. Good Waterman, and I'm sorry I don't have an answer for you directly. So we do know if it is compromised, then we do get immune system actually coming in and producing an immune response against the sperm. So here we've got this a junction between, the transition from our seminiferous epithelium, seminiferous tubule, to our straight tubule. So a straight tubule is basically just the nurse cells or the Sertoli cells, whereas the seminiferous epithelium, obviously we've got our different stages of sperm development here. So we sort of transition here from seminiferous tubules to the straight tubules, which is just basically an epithelium of nurse cells, and then you can see them here, and eventually they then become part of the Rete-testis. So we can actually see some of the Rete-testis out here. Now the Rete-testis is just a simple cuboidal type of epithelium. We often describe them as having some cilia here that help to move the sperm through the Rete-testis here as well. And then eventually these Rete-testis eventually become efferent ductules. Now, as I mentioned, I don't have a nice section of efferent ductules here, but I do have a picture of what they look like. So this is a micrograph I took of one of our slides of the efferent ductules. The efferent ductules, as I mentioned, are the main site for testicular absorption, so testicular fluid absorption, I should say. And they have this very characteristic waviness to their epithelium. And this waviness is because we actually have sort of two main cell types. We have sort of a principal cell that have the microvilli, and they're responsible for the absorption of the testicular fluid. And then we have basal cells. And so because they're different in height, one's sort of a columnar shape and the other is a small, very short cuboidal type of shape, we have this variation in the appearance of the epithelium. So it has this very lovely wavy type of appearance. You might also notice that it's very acidophilic on the outside. These also have smooth muscle, which is contractile, just sort of an autonomic contractility, which helps to move the sperm through as well. So if you ever get a picture in an exam and you're not sure what you're looking at, look for the waviness. That's very typical of the efferent ductules. So there's a number of these efferent ductules, up to 14 or so of these with each testis, and then eventually they coalesce to form the one large, highly convoluted epididymis, which sits on the posterior surface of the testis. So we can see some of the epididymis here. Now the epididymis is sort of divided into three parts. So if we just quickly, so if we've got our testis here, we've got our mediastinum of the testis, and then we have the so we sort of imagine it a little bit like that. So here's our testis. Here's the mediastinum of the testis. This is our epididymis here. I'm drawing with the mouse here, which is why it's quite gross. And so we sort of divide it into three regions. So we have the head of the epididymis, we have the body, and then we have the tail. And the tail becomes continuous with the ductus deferens, which is what this is at here. Okay, so this sits on the posterior surface. So this is anterior, this is posterior. So on the posterior surface of every male's testis is the epididymis, and something which males should be aware of. Obviously, checking for testicular cancer actually involves rolling the testis between your fingers and feeling for lumps and things like that. And just being aware that the normal lumps that you may feel on the posterior surface are the epididymis there. Now, we have moved from the Rete testis to the efferent ductules, which eventually become one large, highly coiled structure, which is the ductus epididymis. It changes in appearance from head to body to tail. We're modifying the sperm in the head and the body regions, and then in the tail, we're eventually becoming continuous with the ductus deferens. So it's more of a storage site. Most of the modification that happens to sperm, this idea of post-testicular maturation, occurs in the upper parts of the epididymis, and then the tail's about storage that then becomes continuous with the ductus deferens. So it's a very unique epithelium. It's a pseudostratified columnar epithelium, and it's one of the few sites in the human body that has stereocilia. Now, stereocilia, which are what these structures are here, stereocilia are very, very long microvilli. So they're immotile. So unlike cilia, which have the motor proteins that helps the microtubules move around, these are actually very long microvilli that have a core of microfilaments, but what they do have on their surface are enzymes. So they're kind of like a brush border of the small intestine by having enzymes on their surface. Sorry, what did the M in the diagram stand for again? Yes, that was the mediastinum of the testis, yeah. Thank you very much for answering that question, Histo Lover. Tarini, I'm glad you got your answer. So the sperm migrate through the epididymis, and as they do, they bypass or brush against the enzymes on the surface of the stereocilia, and the enzymes modify the sperm surface molecules and actually expose new sites that the sperm then can use to help travel through the female reproductive tract and bind to the oocyte, egg coats, and so on. They also are secretory, so they release something like a forward motility protein, which helps the sperm to actually gain their motility. So sperm actually gain their motility as they migrate through the epididymis. It takes about 12 days or so for them to migrate through the epididymis. And they become more and more concentrated. So whatever testicular fluid's left over after the efferent ductules continues to be reabsorbed here, and the sperm become more and more concentrated. One way to identify a nice section of the head of the body region of the epididymis is to look how regular this epithelium is. Look at that nice round lumen. Very, very regular and height epithelium. Very characteristic of the epididymis there. Epididymis has stereocilia, microvilli, or both. It's predominantly stereocilia. The cells which have the stereocilia are referred to as principal cells. They're the columnar cells. We also have basal cells, of course, which are stem cells which replace these principal cells. But primarily it's just the stereocilia that they have. So there is some smooth muscle in the wall on the outside here. Initially that's just 1 to 2 layers, but then by the time we reach the tail end of the epididymis, it actually gains a 3rd layer, which is then continuous with the 3 layers of the smooth muscle of the ductus deferens. So you can see some of the sperm here. They're all quite highly concentrated. We've got some other epithelial cells out here. Some of these would be also some of the residual bodies. That's the so you know when the sperm actually undergo spermatogenesis, they transform that big round cell to eventually the small spermatozoon shape. As they do, there's a little bit of excess cytoplasm that takes place. That becomes known as a residual body, and sometimes that's shed either early and the nurse cells phagocytose it, but sometimes it's not shed until a little bit later during transit during the epididymis. And so sometimes you may have these round spheres of cytoplasm that are the residual bodies of those sperm, continuing and finishing that spermatogenesis process. All right, so this is the epididymis. We do have a nice section, and I might need to just go back to our slides to work out where that, if I've included that section. It might be in this one. So please bear with me. I think I've got a section where we've actually got, okay. So this is, again, a little bit of an unusual section. We've got 2 sections on this slide. One is the spermatic cord on the right hand side here, and this is the ductus deferens. Over here on the left, it's a little bit of a unique section. We've got the testis here, so we've got seminiferous tubules, we've got some of the epididymis here, and then we've got the tail end here. So I think this is probably the head, and this is the tail, and then we've got a little bit of the very early ductus efferens over here. I do know students had a little bit of difficulty interpreting this slide when in our prac class. So let's have a look at this one here. Again, we've got a nice tall pseudostratified columnar epithelium, lots of stereocilia. You can see our sperm highly concentrated in this region here. Relatively thin, smooth muscle layer on the outside. But if we now sort of go over to the other side, this is what the tail region of the epididymis looks like. You can see the sperm a lot more highly concentrated, filling this space. The epithelial height is much shorter. There's very little evidence of stereocilia in the tail region of the epididymis because this is really largely just a storage site. And we can see it's not as evident as you might expect, but we can see quite a thicker, smooth muscle layer around the outside of the tail region of the epididymis. We sort of zoom out a little bit. I want to go over to this section over here. This is actually very early ductus deferens. We can see it's not quite the nice round shape that we associate with the ductus deferens, which to me indicates it's very early, sort of that transition from tail end of the epididymis to the ductus deferens. But we've got our pseudostratified epithelium here with short stereocilia, and then we've got our sperm concentrated in the middle here. but we've got our muscle wall. If we move over to the other side, so this is our spermatic cord. So from the tail end of the epididymis becomes the ductus deferens and runs up within the spermatic cord. We talked about it containing the pampiniform plexus of veins, so that's some of what we're seeing here, some of these plexus of veins, and then over here we've got our ductus deferens. Okay, so we've got our small folded lumen, we've got a lamina propria, and then we have a muscularis consisting of 3 layers of smooth muscle, an inner longitudinal, middle circular, outer longitudinal layer of smooth muscle, which is sort of typical of the ductus epididymis. And if you might remember, we have a similar arrangement of smooth muscle in the ureter. The distal ureter also has an innermost longitudinal, middle circular, outer longitudinal layer of muscle. Now, because this is the spermatic cord, we've actually got the cremaster muscle on the outside here. You can actually see the skeletal muscle here, forming the wall of the spermatic cord there as well. Gregorian 2002, hello. Thanks for coming along. It's great to have you here. Thank you very much also for the follow. Really appreciate you following us. All right. I think that's all I really wanted to say about the ductus deferens, I think. Do we have it? I think we've got another slide here. This is again the spermatic cord. Yeah. So here we've got the ductus deferens, and then we've got our blood vessels. So we've got some of the muscle of the cremaster muscle out here, and so here's the ductus deferens. The preservation isn't that great here. And so here's our epithelium, here's our lamina propria, and then our muscle wall, very, very thick muscular wall. And as I mentioned, it's under the control of the sympathetic nervous system. So during ejaculation, during the emission phase, it stimulates this muscle to forcefully move the sperm into the ejaculatory duct and then into the prostatic urethra. All right, so then there's some of these large blood vessels of the pampiniform plexus and the spermatic arteries and so on. All right, what else do we need to look at? So that's epididymis, spermatic cord. All right, so then this is a resin section. I don't have a very nice paraffin section of the seminiferous, seminal vesicles, I should say, sorry. The seminal vesicles are characterized by having this highly folded mucosa. This is a resin section, so it is a little bit complicated to work out what we're looking at. Again, certainly my CAM 202 students, I wouldn't expect you to identify this section or anything, but we actually got a little bit of retrograde movement of sperm into the seminal vesicles, because we can see sperm here in the lumen. And of course, they're usually separated by the ejaculatory duct and so on. So the seminal vesicles are an important contributor to the structure of seminal fluid. They make up about 65% or more of the volume of semen actually comes from the seminal vesicles. They have smooth muscle in their wall, which is responsive to the sympathetic nervous system during ejaculation. And they contribute things like fructose, which is an energy source for the mitochondria of the sperm, helps them to traverse the female reproductive tract, whereas the tunica albuginea. On the outside here, there's connective tissue. There's a tunica albuginea in the testis, so if you've just come along, you might have missed us talking about the tunica albuginea in the testis. There's also a tunica albuginea in the penis, which we'll talk about in a little while. Yeah, so we do find tunica albuginea in a number of different parts of the male reproductive tract. Remember, we also saw it in the ovary, the tunica albuginea. So tunica albuginea in the testis, Gregorian, I'm not sure if we use that term in the seminal vesicles or not. You might have to look at a textbook to determine that. So fructose is one of the secretions of the seminal vesicles. It also secretes prostaglandins, which help stimulate swimming by the sperm, and it also secretes fibrinogen, which is a clotting protein, a clotting enzyme that helps to clot the semen after ejaculation to keep the semen in the upper reaches of the vagina. So those are the major secretions. It's an alkaline fluid, so bicarbonate's part of the secretion there. That helps to neutralize the acidity of the vagina and protect the sperm from the acidic nature of the vagina, you know, that lactic acid we were talking about in the last stream. This is the prostate gland. This is the prostatic urethra. So it has this sort of upside down U shape. We call this part here, the top of it here, is known as the urethral crest. And then these two spaces down the side, these are known as the urethral sinuses. And you can see different glands emptying into the different regions here. So the prostate gland is extremely acidophilic. So look for that if you get a section, for example, in our practical or our applied exam, look for how acidophilic it is. And that's because the stroma of the prostate gland has lots of smooth muscle and dense connective tissue, both of which are protein, both of which are going, of course, to attract the ears and stains. So this is all smooth muscle in the wall of the prostate gland. And of course, that's under sympathetic control as well during ejaculation. Oops, I'm having a bit of trouble getting the right magnification here to describe the structure. Stay on target. Okay, that's good enough. All right, so this is the urethra, the prostatic urethra, and we sort of divide the glands into a mucosal zone, a submucosal zone, and then what we call main prostatic glands. Now, the main prostatic glands and the submucosal glands empty their secretions into these urethral sinuses, so these little side parts of the urethra, whereas the mucosal prostatic glands empty directly into the urethral crest, into this region here. So you can see the gland structure here. Again, I would not ask my students to actually histologically identify the epithelial type here, but be aware of its nature and its contribution to semen. So it doesn't contribute, somewhere around about 10 to 20% of the volume of semen comes from the prostate gland. Good watermelon asks, the mucosa of the seminal vesicles are highly folded, that's going to be distended when storing the seminal fluid. I'm not sure why it's highly folded. All I know is that histologically, the seminal vesicles can be mistaken for the uterine tube. So remember yesterday's stream we talked about the highly folded mucosa of the ampulla in particular of the uterine tube. It does look very similar histologically in a paraffin section to the uterine tube. It's often one of these ones which we call a lookalike. So the seminal vesicles may look a little bit like the uterine tube. Now, I'm assuming that the folding is more related to increasing surface area for the secretions that it produces, not necessarily about the storage, but it may have something to do with the storage. It's likely to be the production and then maybe a little bit of storage as well. The prostate glands secrete things like citric acid, which actually gives an overall acidity to the prostatic fluid. Citric acid, of course, is an energy source, the citric acid cycle for mitochondria. It also secretes fibrinolysin. So it actually secretes the enzyme which reverses the fibrinogen which is being produced by the seminal vesicles. So after a while, the first thing that happens is the semen clots, and then after a while it breaks down and becomes not clotted, becomes runny, and then that allows the sperm to swim through along the surface of the reproductive tract. So it contributes those two components to the seminal fluid. So we get the sperm from the ductus deferens, we get the seminal fluid, the fructose, the prostaglandins, the fibrinogen from the seminal vesicles. We get a bit of contribution from the prostate in terms of citric acid and fibrinolysin. And then the third accessory sex gland is known as the bulbourethral gland. Now, the bulbourethral gland doesn't contribute to the structure of semen, but more contributes a lubricating mucus onto the head of the penis to help with the movement of the penile head, the foreskin over the surface of the penile head. It's under the stimulation of the parasympathetic nervous system. So we know that erection is brought about by the parasympathetic nervous system, causing relaxation of the muscles within the blood vessels of the erectile tissues. The parasympathetic nervous system, like in the digestive system, stimulates the glands. And so in this case, it's stimulating the glandular secretions of the bulbourethral or the Cowper's glands to provide that lubricating mucus. Is the main energy source of sperm citric acid or fructose? Well, because the seminal fluid, the majority of the volume comes from the seminal vesicles, I imagine that fructose is the primary source, and then citric acid is an alternative source. But because the volume size, good watermelon, I imagine that the fructose is the primary source. All right. So again, this is just another section of the prostate gland. We can see the urethra here. So we can see these main prostatic glands on the outside here. Again, very acidophilic tissue. I don't know if we'll see any of those prostatic concretions, which we can sometimes see in the lumen. With age, often what happens is you can get these little accumulations of prostatic concretions within the glands. It's actually a really good way to identify a section of the prostatic gland. This one doesn't look like it actually has many of those present, but they tend to accumulate with age. I don't think they generally cause any major problems, but they can be a histological feature of the prostate gland. I don't think we saw them in this previous section. Is that one there? Yeah. So this sort of very acidophilic round structure in the glands is known as a prostatic concretion. It's often a good way to identify a section of the prostate, to look for those concretions. Just going to allow this. Can you see the utricle in histosections? I haven't seen it personally. So I'm not exactly sure whether you can see that histo. I'm sorry. Gregorian asked, the stuff that interests me due to the fact that I had to have a testicle removed due to testicular torsion when I was 15. I'm sorry to hear that, but certainly understanding about the structure is one of those things. Often people find something personal, and that may be the reason why they demonstrate an interest in these sort of things. And of course, we can actually get on with just one testis, like we can get on with one kidney. The body has a nice little bit of redundancy there. Here they are, look. So this is what we refer to as a prostatic concretion, very acidophilic. They often have these layers, which we call lamellae. I did see another one over here. Where is it? Here. Yeah, so there's another one here. So that's a prostatic concretion, and it's very classic in the lumen of the prostate gland. So if you can see those, then that's a good indication you're looking at a section of the prostate. What actually are prostatic concretions? That's a very good question. I'm not 100% sure exactly what they are. A lot of the time these things tend to be acids or cholesterol based, but I'm not 100% sure. I'm sorry, I can't ask that. Gregorian asks, is this website or database free to use? At the moment, it's not. We are currently in the process of developing our own virtual microscopy system, which we hope to have available early next year. If you are an academic, then these slides are actually on the virtual microscopy database, which is run by the American Association of Anatomists. But at this stage, I'm sorry, they're not available to the general public, but they will be. I promise that they will be. And hopefully then, if I'm doing some streams next year, you'll be able to follow along. As I look at my slides, you can be looking at your own slides. And that's That's our dream anyway. So hopefully we can get there. I do notice the time, we're almost close to the end of the stream. I do want to quickly just have a look at the structure of the penis here. We know that in cross-section the penis is composed of two upper erectile tissue masses known as the corpus cavernosa, and then we have a lower erectile tissue mass known as the corpus spongiosum. Yeah, I'm sorry Gregorian, I'm sorry it's not available for everyone at this point in time. This is the corpus cavernosa that contains the erectile tissue masses here. So here we have the vascular spaces or lacunae, and they have smooth muscle in their walls. Housed within here, we would have the main arteries, the deep arteries of the penis, and then we have a thick connective tissue, tunica albuginea, on the outside of each of these erectile tissue masses. In the periphery, we would find the veins of the penis, and then down the bottom here, we've got our corpus spongiosum, which contains our urethra and the erectile tissue masses. You can see that the tunica albuginea is much thinner in the corpus spongiosum than it is in the the corpora cavernosa here, because of course it's about trapping blood and keeping things under hydrostatic pressure, so that maintains the erectile tissue, maintains the erection. If you actually have that sort of hydrostatic pressure in the spongy urethra, around the spongy urethra, then you may impede ejaculation and so close off the lumen. So we don't have as well developed tunica albuginea around the outside of the corpus spongiosum. So during erection, what happens is we get stimulation of the parasympathetic nervous system. This causes relaxation of the blood vessels within the corpus cavernosa, which allows more blood to enter. Where is the urethra? This is the urethra here, this thing that looks like it's got teeth, what we call the mouth of... in my lecture, I put up a section of spawn here, so the eyes and the mouth here. So this is the urethra in the corpus spongiosum here. So the parasympathetic nervous system causes vasodilation of the deep artery of the penis, as well as the smooth muscle within these vascular spaces that enables more blood to enter into these spaces. They expand to accommodate that increased volume. As they expand, they begin to press against the veins in the periphery, and so that impedes venous return. So more blood comes in, less blood returns, and eventually this leads to increase in size of these erectile tissue masses, leading to an erection. Yeah, it does look a little bit like he's looking into your soul. It looks a little bit like someone screaming or something. It's quite terrifying, this section. That's the erection. We talked about ejaculation consisting of those two components, the emission phase by the sympathetic nervous system, the ejection phase by the lumbar reflex, the somatic lumbar reflex there. All right, well, that's pretty much it for the male reproductive tract, male reproductive system. So that pretty much concludes our sort of tutorial-based type of streams. What I was thinking of doing was to do a little quiz overview revision session of the entire semester's work that we've looked at here at the University of Tasmania. So looking at the digestive system, looking at the urinary system, looking at the two reproductive systems, and having sort of quiz-based, micrograph-based, Kahoot-based questions as part of your revision. So I was hoping to run that next week sometime during study week for those of you who might be interested. Otherwise, if you've got any other questions, we can also talk about those in a future stream or something as well. Good Watermelon, you're very welcome. Hopefully you found this useful. I had fun talking about it today. It's really great that everyone's come along. Tarani, thank you. Histolover, excellent. I'm glad you'd be interested in something like that. Last year I ran an online version. Actually, I ran two because everyone liked it so much and we never finished everything. So we actually ran a second session And we got a lot of the students from the CAM 202 group to come along. So we had like almost 100 students participating. So if we could get anywhere near that, that would be awesome. Histo 1, 2, 3, 4, 5, 6, 7. Thank you so much for the live streams. They've changed the game, to be honest. I wish we had these for all subjects. Helps learning so much. Oh, that's really awesome. Thank you very much for that lovely, kind words. So that does it for this stream. I hope you found that useful. We'll see you in the next streams. Keep an eye on my Twitter. For those of you who are CAM 202 students, keep an eye on the announcements on Milo. I'll make an announcement about when we'll be running the stream. It'll probably be a couple of hours long, just so we can cover it all. You can come ask questions. We can talk about things. So I hope you enjoyed that stream. I'll see you in the next stream. Have a great weekend, and I'll talk to you again soon. Hooroo!